Hello and welcome to Dunia's Alchemy. This video is going to be on microscopes. This is a GCSE Biology topic and the video breakdown is below. The first thing we're going to have a look at is light and electron microscopes, followed by magnification formula and units, and lastly microscopy required practical. So let's begin. We learned about cells and their organelles in the last video, but since they cannot be seen with the naked eye, we have to use powerful lenses to be able to identify them. Two such examples are light and electron microscopes. Before I go into the differences between the two, look at these simplistic images and try to find any differences before I share. Feel free to pause the video. So let's see if what you identified compares with the advantages and disadvantages of both the light and electron microscope. So let's begin. The advantages of a light microscope is that it's cheap, it's portable, so very easy to carry around, it's easy to use, and you can view living and dead specimen. Disadvantages of light microscope is that it generates 2D images, so you can't see internal structures easily. It also has a lower magnification and a lower resolution. The advantages of an electron microscope is that you can see 3D images. It has a higher magnification and a higher resolution. However, the disadvantages of electron microscope is that it's expensive, it's large and not portable, requires special training to operate, and it can't view living specimen. So let's give a pause here for a minute. Do you notice a pattern? So if you look at the advantages of the light microscope and we compare them with the disadvantages of the electron microscope, you can pretty much see that they're opposites. So while one is cheap, the other's expensive. While one is portable, the other one's not portable. While one can see living and dead specimen, the other one can't. So essentially, I would advise, try to learn one and you'll be able to figure out the other one. And the same happens with the electron advantages versus the disadvantages. So it generates 3D images versus 2D images, high magnification and resolution versus low magnification and resolution. So that's a really nice trick because I do see this come up as a 6 marker. Let's have a look at the magnification formula. A really simple way to remember this is I am. That's literally how I remember it. I call it I am. So I over here means your image size. This is usually in millimeters or micrometers. A is actual size, and similar to the image size, you tend to find them in exam questions in millimeters or micrometers. And then finally, M stands for magnification. There's no units for the magnification because your image size and your actual size should have the same units and therefore cancel out. Let's try a question. Try to rearrange and make M the subject. Feel free to pause the video. No, I have not gone MIA on you, but that is actually the answer. All you need to do is cover up the one you're looking for. In our case, that's the M. And we're left with I divided by A. M-I-A. Let's try this question. A student observes a plant cell under a light microscope. The actual length of the cell is 0.02 centimeters. On the microscope image, the cell appears to be 6 centimeters long. Calculate the magnification. The magnification is 6 divided by 0.02 centimeters, and that should give you 300. Note how there are no units, and sometimes you can just write across at the end. Over the years, I have seen a lot of students struggling with their conversion. So I've given you this over here. Feel free to copy it down into your books, and we'll go over it. So meters to centimeters, centimeters to millimeters, millimeter to micrometer, and then finally to nanometer. So this is going in a descending order. Now, as you can see, we're multiplying each time, which means if you were asked or prompted to go in the opposite direction, so for example, nanometer to micrometer, you'd simply do the opposite, which is divide by 1,000. However, what if you were instead asked to do micrometer to centimeter? In that case, as you can see, going from micrometer to millimeter is 1,000, and then going from millimeter to centimeter, a factor of 10. In total, that's about 4 zeros. So what you do is you divide, instead of timesing, because we're going backwards, you divide by 10,000. That's your 4 zeros. So let's try some questions on this. Convert the following. Feel free to pause the video and have a go at these. The answers are now on the side. Give it a score out of 6. Did you get all of them right? Here, have some flowers. You deserve it. Let's have a look at this 6-mark microscopy required practical. This is for when they ask you about the method. So let's begin. Using tweezers, peel a thin layer of epidermal tissue from an onion. This is so that light can pass through. Step 2. Place the tissue flat on a glass slide. ensuring there are no folds or overlaps. Step 3, add 1-2 drops of iodine solution. Sometimes you can use methylene blue, but iodine should be fine, to stain the cells. This makes the features such as your nucleus and cell wall more visible. Step 4 is lower the cover slip over the specimen using a mounted needle, ensuring there are no air bubbles trapped and can obscure the view. Step 5. Place the slide onto the stage and secure it with stage clips. Starting from the lower power objective lens, use the coarse focus knob to bring the stage up until the specimen is nearly in focus. Look through the eyepiece lens and slowly adjust the coarse, then fine focus knob until a sharp image is seen. And finally, increase the magnification by switching to a higher power objective lens, then refocus using the fine focus knob to observe cell details like nucleus, cytoplasm and cell wall. As a bonus, you can also add your IAM calculations at the bottom when you're talking about this required practical. This is our video complete. We have now gone over light and electron microscopes, magnification formula in units, and microscopy required practical. Thank you for watching, and make sure you do the 3-2-1, share it with 3 others, 2 clicks, subscribe and like, and finally 1 comment. Have a great one.